Hey guys, so today we're going to be talking about the last part of chapter 6, worked our way from the nucleus all the way to the outside of the cell. So this is going to take us to the cytoskeleton and the extracellular matrix. So we'll go ahead and start with the cytoskeleton. So there are three main elements of the cytoskeleton that we're going to talk about, and each one has its own function and structure. So we'll be talking today about microtubules, which are going to be composed of tubulin proteins, microfilaments, which you may also know as actin filaments, which are going to be composed of actin, and intermediate filaments, which we'll see can be composed of a variety of different proteins. So we'll start with microtubules. So microtubules are the largest of the cytoskeletal elements. They're involved with a wide variety of cellular processes, including chromosome movement during mitosis, meiosis, vesicle movement within the cell, providing structural support, and we'll see an example where they help the formation of cilia and flagella for whole cell movement. So let's start at sort of the beginning organization center for microtubules. In animal cells, this is the centrosome. So microtubules are organized at the centrosome. The centrosome is this entire sort of yellow fuzzy bit and the pair of centrioles. So centriole 1, centriole 2, and this cloud contains protein complexes that are useful for assembling microtubules. So think like pieces of tubulin and other proteins that are going to be helpful for putting these together. This is kind of like if you dump all your Legos out on the floor, you have a structure that you've made, but surrounding it are all those Legos still just waiting there to be assembled. Each centriole is made of 27 microtubules. We'll see more about the detailed structure of these microtubules in a minute, but you can see here that they're organized in a 9x3 structure. So each one of these hollow tubes is a microtubule. You can see they come paired in groups of 3, and there are 9 of those groups of 3. So this is 27 total microtubules, and we call this a 9x3 structure. Plant cells do not have centrosomes, but they definitively do have microtubules. They just organize them a little bit differently. So let's go ahead and look at the structure in depth of a microtubule. So here's a microtubule. We're really close up. You can see that from the smallest unit, these are composed of what are called tubulin dimers. Tubulin is a protein. It's synthesized by a ribosome. We have two main types of tubulin here, though other varieties exist. This is alpha tubulin in red and beta tubulin in yellow. These almost always already exist in dimers before they're added to the structure. These dimers then self-assemble into strands. So each one of these strands are called protofilaments. 13 of these strands, so counting 1, 2, 3, all the way around this ring, come together to form a tube that's hollow in the center. So here we're looking at it straight on. You can see that there are 13 of these. Okay, so 13 protofilament strands. And this altogether is a microtubule. So these are fairly large in structure relative to the other cytoskeletal elements. You can see formed a hollow tube. Okay, so let's talk about the way that microtubules form. It turns out that microtubules are very dynamic in the cell, and they have to be able to change length really rapidly. So we can see here our familiar friends from the previous slide, alpha tubulin and beta tubulin, and you may notice a couple of other things going on here. So these proteins are bound to small molecule called guanosine triphosphate, or GTP. This is the same guanosine that's present in DNA, and it's similar in a lot of ways to ATP. It's going to be, instead of adenine, we've got guanine here, and 3 phosphates. That's the triphosphate. So these tubulin proteins are bound to a tiny GTP molecule. Okay, it's very small relative to the protein. And when they're bound to this GTP, they are very stable for docking and adding on and forming protofilaments. So here we're going to have GTP bound tubulin in red, so specifically the beta tubulin. Whenever it's red, we can see that we're going to have our beta tubulin, and as we transition to brown, this is going to be a hydrolyzed form called GDP. So let's explore that a little more. Remembering that GTP is the stable form, over time, this GTP can be hydrolyzed. Okay, so you may notice what's different about this molecule GDP. This is a diphosphate. So we've just lost one of the phosphates. So we start out our stable form bound to a triphosphate form. And here, once it hydrolyzes over time, we've got a diphosphate form. Okay, that's just kind of over time, that's what happens. This is the hydrolysis step we're looking at here. The GDP bound form is less stable. We haven't seen a picture of this yet. We've only talked about putting it together. But as this hydrolyzes over time, this is going to cause a depolymerization or shortening of the microtubule, which is also called a catastrophe. So let's see that in a little more detail. So you can see in part C here, what we're doing is starting to polymerize. These protofilaments that are bound to the GTP form are going to be kind of zipping up together. So they're actually coming together. Think of the zipper moving up in this example. In part D, we've hydrolyzed to the GDP bound form. So if the hydrolysis catches up before we can add new dimers on, this destabilizes the microtubule and it actually just peels apart like a string cheese. Okay, we'll see an example of this in motion in a couple of slides. So we have this balance between polymerization and depolymerization that is basically dependent on how fast we can continue to stack GTP bound dimers onto this structure. If we can rapidly keep adding, then we keep ahead of the hydrolysis. If we slow down and don't add as quickly, the hydrolysis catches up and we can end up in catastrophe. And this sounds really random, but it's actually highly regulated by the cell, mostly by controlling the ability of the cell to have access or the availability of these GTP bound forms. Okay, so we're just remembering that the GTP bound form is stable. So let's add one more detail onto this and see how it looks in a cycle. So we've only been looking at sort of the growing end in our last couple of examples. The end which we can actively add and subtract is referred to as the plus end. Okay, so there's some directionality. This will come in handy in a little bit when we start talking about motor proteins. The stable end, which doesn't usually grow, here you can see we've actually even capped it up with different types of tubulin, but it's just going to be our stable end. It's not going to be rapidly growing. This is called the minus end. So we really don't have to worry about what's going on here. We just need to know that's the name of it. And we can focus for now on the plus end, where things are going to happen. So again, you can see this sort of cycle between polymerization and depolymerization, which we also call rescue and catastrophe. So polymerization, again, think of the zipper moving up. We're zipping up all of these 13 protofilaments so that they form a tube. And again, this is controlled by the amount of GTP-bound tubulin dimers. Okay, so the ones that still have GTP, if we can add them quickly, if there are a lot of them available, then we tend to lean towards polymerization. So there's a lot of biochemistry at work here. If we let this hydrolysis to the GDP bound form catch up, we peel apart like a string cheese. And again, this is highly controlled. It turns out that the cell can really balance this, particularly when it needs to maintain an approximate length of a microtubule. So we can kind of treadmill back and forth between catastrophe and what we call rescue, which is where we'll start adding again. So it's almost kind of like Jenga a little bit within the cell, kind of controlling whether we're adding, subtracting, building onto the top, or whether it falls down. And it seems like catastrophe is a really bad thing, but it's very important. It's just as important as polymerization. We have to be able to shorten the microtubules just as much as we need to polymerize them. So a good example of this is during mitosis, when chromosomes are pulled apart. It looks like we just kind of reel in the microtubules, but in actuality, the microtubules are shortening. And that's how we're able to separate those chromosomes. So let's look at an animated example of what's going on here. Okay, so we're going to see our GTP dimer being added. That's going to be in red. The hydrolysis happening in blue, and you can see we're just keeping up with it. If we stop adding, we hydrolyze everything and pull apart those protofilaments. So you can see it's a pretty distinct structure. Let's just watch it one more time. Again, adding, 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 hydrolysis, keeping just one step behind, catches up, and we're destabilized. So again, it's very important that we're able to regulate which of those phases we're in. Okay, so now let's talk about a couple of other purposes for microtubules. We've talked about how they're actually going to be built, the components that they're made of, but there's going to be a couple of ways we can use these. Okay, one of the things that we mentioned at the beginning of the microtubule section was that they're going to help us form flagella and cilia. So flagella and cilia, as we know, these are forms of movement for the cell. So you can think a good example of flagella is going to be on sperm cells. This is going to be a long tail-like structure. Cilia are going to be shorter, and you can think of cells as having many cilia that kind of beat in a power stroke motion that help propel the cell. So both of these structures are formed similarly from an arrangement of microtubules. Okay, so we can actually see in this image, this is a TEM that we're looking at, transmission electron image. You can see that this is the plasma membrane, and we've got microtubule structures that start embedded below the surface of the plasma membrane and extend up into the structure. Okay, so the base of this anchorage in the cell, is referred to as a basal body. And we can see this is a basal body, and it should look really familiar to you. So where else have we seen that 9x3 arrangement, okay, made of 27 microtubules? This is very similarly structured to our centrioles. Okay, you can see some other types of proteins kind of holding this together in the center. Okay, similar 9x3 arrangement. The basal body is going to act like an anchor in the cell to hold things in. You can see how it extends well below the cell surface. Above the plasma membrane, kind of up here at this level, we convert to a 9x2 with 2 in the center arrangement. So we've got 9 sort of units around. We've also got them in pairs here. Okay, and this is a really cool image. You can actually see the tops of all the protofilaments, those tubulin dimers, really emphasizing how powerful electron microscopy can be. Okay, and we've also got a pair in the center. So something that's going to make this a little bit special, that's going to allow for movement, is the addition of motor proteins. So you guys have hopefully heard of motor proteins before. Probably myosin's the most popular, but we're not ready to get to myosin just yet. Instead, we're going to be looking at a motor protein, which is, again, as the name suggests, going to help us with movement. We're now going to be dealing with one called dynein. Okay, so this is a dynein motor protein. We'll see how this moves in a couple of slides, but for now, we're looking at how these are going to be linked. Okay, so first, let's just look at the way dynein moves along a microtubule. Okay, we'll go ahead and connect them up to our cilia and flagella in a bit, but let's just get used to the idea of dynein. So dynein is cute. It does look like this structure where it's got sort of a head and then 2 legs. So you can see in this video we're about to watch, here are the 2 legs, looking good, dynein. Okay, and here is sort of the head of this. Okay, the head is generally used to interact with things like microtubules or vesicles. And this has a fairly strong attachment to whatever it's holding on to. Think about your hand gripping something that you're carrying. Whereas the feet are going to have sort of a more transient attraction to the microtubules, which will allow us to walk. So think again, just like your feet lifting off the ground. Your feet are not permanently attached to the ground. So let's watch this guy go. So you can see this is actually the way they move. They use ATP to power their movements, but they literally walk in kind of a wonky, drunken way, but almost just like a person. Okay, and again, if you could imagine something being attached to the top of this. So this is a pretty cool image. Okay, so let's add this back in to our idea of flagella and cilia. So here are some doublets. We're not quite at the complete cilia flagella structure just yet, but let's imagine this is the head of our dynein molecules. It's attached pretty strongly to this top pair of microtubule doublets, okay? Whereas the feet can walk along, okay? So our feet can walk along. If we don't attach these microtubule doublets to anything, these guys can literally living style just sort of walk off the edge of the second doublet. So think of it kind of like living style, just walking off the edge, okay? But we know that's not the way that cilia and flagella work. We don't want the microtubules to slide past each other. We want them to bend to create motion. So that's where our basal body and other anchorings are going to come in handy. So when we do get to the point where we're going to be looking at this sort of swaying or power stroke movement, we have our same doublets with our dynein attachments and more permanent head attachment and this transient walking foot attachment. We anchor this in the cell with the basal body and other anchoring proteins, kind of scaffold proteins cross-linking, you can see here. When these feet try to walk, instead of sliding, they actually attempt to walk, but they instead create a bend, okay? When they relax, we can then bend the other way. So this is a cycle of walking, detachment, resetting, walking, detachment, resetting, that causes our cilia or flagella to bend in the way that it needs to. In cilia, this is more of a power stroke type movement. With flagella, we end up with that very characteristic undulating motion that we see in videos of, again, the motion of sperm is a very good example. So this is one of the ways that we can contribute to whole cell motion, thinking about powering the whole cell through space. But it turns out microtubules are very important for actually moving things in the inside of the cell as well. Okay, so in the last couple of videos, we looked at the endomembrane system and we talked about vesicle motion in the cell. And we talked about how these vesicles can butt off from places like the ER or the Golgi apparatus. They can make it to the endosomal lysosome pathway to the plasma membrane. Alternatively, we can bring things in from the plasma membrane through endocytosis. So there are a lot of ways to move vesicles within the cell. Up until now, it's looked like these things just float to where they need to go. But in reality, they're carried. Okay, so I kind of think of this like the way that you might imagine if you've seen the movie Up, guy walking, okay, and they've got the house supported by lots of balloons, so this would be the house, kind of tied to him and just walking along. The house isn't just floating aimlessly, it's being kind of dragged along by a person. So that's the way you can think about vesicle traffic in reality. So these things have very set highways that they can go on. And see, here's a motor protein that will allow us to travel, attach the head to our vesicle. Here are some vesicles in the actual cell or microtubules, and you can bet that buried up underneath here we'll have some motor proteins as well. So our microtubules act like highways to allow for vesicle transport. And again, they don't just float around, they're pulled along by motor proteins. Most notably, dynein and kinesin. And we'll talk more about kinesin and compare it to dynein in a moment. But let's look at a video of vesicle traffic. So this is a pretty cool video that's widely popular to show vesicle transport. And you can see a motor protein. My opinion is that this is kinesin. Actually, it says it in the top. And you can see this little guy having that same foot motion that our dynein had while the head is attached to the vesicle. So there it goes, attached to the receptor. And so you see one in the background. These guys aren't just floating randomly. They attach to a microtubule track and are kind of dragged along to whichever direction they need to go. So it turns out, again, if we need to think about directionality, you may remember that some things travel more towards the inside of the cell, and some things move more away from the inside of the cell. So motor proteins have directionality. They cannot walk both ways along the microtubule. They have one way that they can go, and they have to stick to it. So if you need to go a certain direction, you need a motor protein that's going that way. This is a very large generality because there are lots of different types of dynein and lots of different types of kinesin, but in general, many dyneins tend to move more towards the minus end of the microtubule. So that's that capped end that isn't growing quickly. You can think of this in a way as sort of moving towards the inside of the cell, okay? It's not always true. It depends on exactly where the minus end is. But generally speaking, you'd be moving more inward. Kinesins, on the other hand, a large number of those tend to move towards the plus end or the actively growing end of the microtubule, which often corresponds to the more outer direction. So in general, dyneins can move sort of more towards the centrosome, while kinesins in general may move away from it towards the plasma membrane. So if we think about tying that back in to things like K-tail retrieval, this is a nice image, both because we've seen it before and because it shows sort of a multi-directional kind of trafficking here. Before, again, we were thinking of vesicles as just floating, okay? They just float to where they need to go, but now you can kind of extend the idea of microtubules being just underneath these and motor proteins. And you can think about the types of motor proteins that might pull things from sort of the outer stacks of the Golgi more towards the inside of the cell, and things that might promote a forward motion through the Golgi and then eventually out towards wherever it's going after that. So I think this adds sort of a nice layer to something that we've seen before. So we've talked a lot about microtubules, okay? We'll also cover in this video microfilaments, actin filaments, which are actually in a lot of ways similar but yet distinct from microtubules. So actin filaments are going to be the smallest of the cytoskeletal elements. So get comfortable again with calling them microfilaments as well, and don't get them confused with microtubules. So think about a filament as being small and a tubule as being a tube or larger than a filament. Your book and in general scientific talk uses these terms kind of interchangeably, so be comfortable with both. These microfilaments are involved with things like cell surface structure. So they're not the only element, but we'll see that they allow for dynamic changes in cell shape at the surface. So microvilli is something that we'll look at, and then amoeboid movement is something pretty cool to look at as well. And also responsible for movement of the chloroplasts within the plant cell, which is called cytoplasmic streaming. Okay, it doesn't just move chloroplasts, but again, it's easy to see chloroplasts, so we can kind of gauge where they're going. Okay. Obviously, you can't talk about actin without talking about muscle contractions, so we'll be hitting that as well. Okay, so let's start with our microfilaments. All right, so these are assembled, these actin filaments are assembled in a lot of ways, they're similar to microtubules. So they're going to have sort of a minus end where things don't happen as quickly, and a plus end where things are going to, you can see this fat arrow, this means we're really heavily leaning towards polymerization. Okay, whereas it's sort of at an equilibrium at the minus end, so neither adding nor subtracting. You may notice that we've got a more red, ATP bound form, and a more pale, ADP bound form. So like microtubules, we're going to assemble our subunits in a way that's more stable. So these are actin proteins. Notice they're just singular. They're not dimerized the way that our tubulin was, made of actin. When they have ATP bound to them, they tend to polymerize. If hydrolysis catches up, then they tend to depolymerize. So again, this allows for a lot of dynamic lengthening and shortening as needed. But the cell is regulating it very tightly. It's not just random. It knows exactly what size these need to be, and it will make sure that those lengths change appropriately or are stabilized as needed. So we've got the assembly taken care of. You can see in this image, we're going to have some structures contributing to things like microvilli. Okay, so we've got microvilli. These are different than cilia. Don't get them confused. These are going to be projections that allow us to have increased surface area. So you might have these in things like gut epithelial cells that are designed to absorb things. Okay, so like intestinal cells. All right, so each one of these tiny strands is going to be an actin filament, and you can see it's just under the plasma membrane. So I like to think of this as like a hand in a glove, where the glove is going to be the plasma membrane, and the hand are these actin filaments, this bundle that sort of push this up, okay? And microvilli are a fairly permanent structure, again, because we want surface area. We'll talk in a moment about times when we can have a little bit more dynamic motion here. Just up under the surface of the cell, so this will be our plasma membrane border, you can see that we're going to have kind of a network of actin. This is also called a cortex. Okay, we'll see that on the next slide as well. Below that, we've got some intermediate filaments, which we will talk about in this video, but not yet. Okay, so focusing on the actin. So let's look at some ways that actin can contribute to motion. So like our microtubules, they're highly dynamic, and they can do a lot of different things. So on the top, we're looking at the motion of an amoeba. So hopefully you guys have been able to see the way that an amoeba moves. It sort of blebs out one side, attaches, and then pulls the rest of its body in to meet it. Okay, this is also called pseudopodia. So our false foot, sticking a little foot out here, and then just sneaking the rest of its body up behind it. Okay, you can see just up under the surface, this is a network of actin. This is our cortex, which forms kind of a gel. And you can see that the actin is redistributed so that we are pushing out this edge. We're going to adhere to whatever surface with some proteins, probably part of the extracellular matrix, surface proteins. Once we've attached, we just allow this to contract back up and repeat the process. So this is highly dynamic. Amoebas can really get trucking when they need to. Additionally, we can see actin filaments sort of rearranging in a way that creates a current. So if you've ever been like a kid in a pool that wanted to create like a little whirlpool so you kind of walk around and around, that's the kind of current that we're talking about. And this is a very distinct current, and it's called cytoplasmic streaming. So a result of that in plant cells is that we can see chloroplast movement. And again, chloroplasts are really easy to see, and they probably move about the speed that I'm going here. So it's a pretty distinct movement. It's not very slow. Okay, and it can also pull other molecules along with it just to sort of redistribute things that are going along in here. So again, a lot of dynamic motion. Okay, and again, it wouldn't be actin if we didn't talk about muscle contractions. So actin forms the base of the structure that we're going to call the sarcomere. So here are our actin filaments, interspersed peanut butter jelly style with myosin units. Okay, myosin filaments here. And again, myosin is going to be our motor protein. So we have a stable actin filament interspersed with motor protein myosin filaments. You can see that they are composed of units that have some space between them as we're relaxed. This whole unit being referred to as a sarcomere. Okay, so again, I think all of you guys have seen this before, but let's just review it a little bit. Okay, so we're going to go through the steps of a muscle contraction in detail, and then we'll see it in an animated way. So here we've got the myosin head. At this point, we are attached to the actin filament. So we're kind of in a stage where we've probably just finished a contraction and we're getting ready to reset and do it again. You can see minus N plus N, okay, actin filament. Here we've got this attachment. So in order to detach and reset, we need a molecule of ATP. Myosin binding to ATP means it has to let go of this actin filament. So think about its hands being full, and so it has to let go. Okay, notice it's still kind of in this used, not ready to go kind of a posture here. In order to move into what we call the cocked state, where we're obviously ready to repeat the motion, we're going to hydrolyze our ATP to a DP. Notice the third phosphate that we've released is still hanging on, but it's no longer attached to the ADP. Okay, we are a diphosphate. The third phosphate is separate, but still in myosin's hands. This allows us to resume sort of a cocked, ready to go position. As we drop the phosphate, so think about, okay, our hands were full, so we just drop the phosphate. This allows us to again hang on to both the ADP and our actin, okay? So when we let go of the ADP, we can fully devote our attention to this forward motion or the power stroke. So let's see how that happens in a video.
Myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross-bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
So actin and myosin muscle contractions just as fresh as it was in 2009. So now we've talked about several ways that actin can contribute to motion inside the cell. So we've talked about things like cytoplasmic streaming, amoeboid movement, and again, muscle contraction. We've also talked about the way that it can help the architecture at the surface of the cell, including things like rearrangements of the actin cortex and microvilli structures. So now we'll finish up by just briefly introducing intermediate filaments. So our intermediate filaments are kind of, in some ways, almost like a grab bag of filaments, in the sense that they're not one kind of filament in the way that actin and tubulin were, but there are many different proteins, different types that range in size between, somewhere between our actin filaments and our microtubules. Intermediate filaments are also primarily involved with more permanent structures than dynamic ones. So with actin and with tubulin, or microtubules, we see this potential for rapidly changing the length of the structure. Intermediate filaments are more involved with structure we want to keep there. So a notable example includes things like keratin, which we know keratin is found in our skin cells and our hair. If you think about the way that hair tends to persist, it's like difficult to get rid of that. That's the way that keratin is arranged. So we'll look at how it's structured in a moment. But think about permanent structures, things that you don't want to assemble and disassemble rapidly. It's like once you put it together, you want it together, like a piece of IKEA furniture. Okay. Another example of this are the nuclear lamins. So if you remember back to our unit on the nucleus, we talked about the nuclear lamina, which provided support for the nucleus. Lamins are going to be one of the proteins that are in the intermediate filament category, and they form sort of a mesh-like network just below the inner membrane of the nucleus. So let's look again at our intermediate filaments. And again, we saw this picture previously, but we're focused on actin, but you can see that the intermediate filaments, they're larger than our actin and form sort of a mesh-like web below the actin cortex. So let's look at something like keratin and see how it's assembled. So here we have a keratin monomer. So this is one protein that was kind of squirted out from one ribosome. We take two of those together and you can see that they sort of interweave and twist together. This is a dimer. Take two of these dimers, bind them together. So we have 4 keratin proteins. This is a tetramer. We take 8 of these tetramers and kind of arrange them into a sheet. Okay, it's kind of difficult to even keep count here, but you can see tetramer, 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 etc. And the sheet can actually be rolled up like a rope. So you can see that the way that this is going to be assembled is very complex, not easy to undo at all. And so once you make keratin, you mean for keratin to stay there. So this is a highly structural protein. It helps to maintain, again, shape of things inside the cell. There are many different types of this, not just keratin, but this is one of the most notable examples that we always hear about. But you can see just the way that it's composed, that you don't want to be assembling this and disassembling this rapidly. This is going to be something that once you make it, you intend for it to stay around. So in this presentation, we've talked about the 3 main types of cytoskeletal elements: microtubules, which are the largest; microfilaments, which are actin filaments, the smallest of the filaments; and intermediate filaments, which contain things like keratin, things that are intermediate in size and for the most part sort of built to last. So again, our microtubules and microfilaments are largely involved with dynamic motion within the cell or the whole cell in general, while intermediate filaments most of the time provide more permanent support. And in our next video, we'll be traveling all the way to the outside of the cell, the extracellular matrix, and finishing up our tour of the cell in Chapter 6.